Начинать. Уважаемые слушатели, участники нашего семинара, сегодня у нас выступает Игорь Чернявский с продолжением своего доклада про особенности кровообращения и процессов переноса в плаценте человека, часть 2, в скобках на английском языке. Да, язык остается английским. Да? Вы отказываетесь переходить на русский язык.
Я еще раз извиняюсь, я просто думаю, что так будет более гладкая презентация, потому что иначе...
Пожалуйста, это на ваше усмотрение, но моя просьба остается, как и раньше, тогда, если можно, так не торопясь, чтобы наши слушатели могли вас лучше понимать и следить за докладом. Пожалуйста, Игорь, мы вас с интересом слушаем.
Thank you very much, Vitaly Alexeyevich. Thank you, Anastasia. Again, I apologize for presentation in English, and hopefully the audio equipment will cooperate this time. I'm very happy to take questions anytime in Russian if you prefer. So the reason for English presentation mostly is because most of terminology and materials are in English. And I'm not sure of always the right Russian equivalent. So what you see on the screen now is a very recent work, not mine, of course, but just to show that it's very rapidly evolving field. It's literally published a few weeks ago, a study in Nature, which shows that for the first time it's possible to maintain an embryo outside the womb for extended period of time, I think for at least a week, which is quite amazing.
I'm sorry, Igor, we don't see, at least I don't see, probably the others also, the picture. In fact, if you can, okay, this is perfect. Yes, thanks.
Yeah, I just wanted to show you the title. So what you can see on the screen is the embryo of a mouse, mouse embryo, which is filmed outside the womb and being developed. And on the background, there wasn't focus on that study, but on the background, behind the sac, you can see a bit of the placenta, the mice placenta. So you see my cursor. And you see this pulsation is the beat in the heart of the embryo. And they just put it in the context, that is how they've been cultured. So you can see these little balls inside the rotating vials with the nutrients and the oxygen being passed through that vial. So it's literally the embryo growing inside the glass vial. So that shows that it's, and at some point might be possible to really develop a truly artificial womb, which is, the continuous process of development of an embryo. So with that, I will switch to the presentation.
Excuse me, Igor, but it's really so interesting that I'd like to ask you a question. But so you say that it is several weeks ago. So what is the result? So how long the gestation period in mice probably is quite short. So are they born finally, these mice?
No, it's still very immature, of course. No, it's not complete. I would hesitate to quantify the period, but I think they managed to keep it for one more week in this condition. And some of the embryos already were a bit mature, so they're not all of them fully developed in this condition. But I think it's still ongoing work. So I would say it's probably approximately 1/3 of the mice cycle, might be closer to 1/2, but not the full cycle, of course. And actually the limit, that's a good question you asked. The limit of this technology at the moment is that it becoming mature enough and they can no longer be supported by diffusion, which you can see in this vial. They can no longer be supported by diffusion of oxygen, and they needed direct circulation, which of course cannot be provided by this technology. So they now need effectively to be transferred to the artificial placenta in a sense. So they needed the device which can provide direct flow through the placenta rather than just a passive oxygen.
Okay, thanks.
So in a few seconds, I will try to share the slides. Okay, so here they come. So I will very briefly remind you where we stopped last time, and then move on to the second part. So the story here is the story of two circulations. And again, briefly reminded, it's not just my work. There are lots of people who I collaborate with and who worked in our group. So majority of the work is done by my former colleagues, postdoctoral researchers, Alexander Ehrlich and Philip Pierce, who was working on my project. And of course, we have a very fortunate to be working with experimentalists, Win Toon and Helen Bishop, and Gareth Nee, who is now lecturing Chester University. And it's all about trying to understand the mechanics and biomechanics of living systems, tissues. And what I showed you here is more or less that process. Is it? It's not progressing. Can you see any new slides? I think it doesn't seem to be changing.
No, it's the first slide with the title.
Okay. Let me see. Okay, yeah, now it's working.
Yeah,
sorry
now it's okay. Yes.
So what is it I mentioned? That's just to acknowledge again all the colleagues. So Alex Ehrlich and Philip Pierce are the key names here. Okay, so I know if you haven't seen this book, I highly recommend it, but I wouldn't talk about this again. So these are the development. So from Da Vinci, very detailed reconstruction of the womb, to the more recent work on artificial placenta, which allowed to connect the other end of pregnancy, where very immature fetuses, in this case of a lamb fetus, been kept alive for an extended period of time, in this case a month, which is absolutely amazing for that to happen, because it's a very intricate, and because it's it's a blood culture, it's very easy to clot and so on. So they kept it attached directly to the umbilical cord of the lamb, and it couldn't breathe otherwise because it was very mature, and it was arrived very healthy, very happy. So that's totally technology is in rapid development. And what you saw at the beginning is the other end, where you can start culturing the embryo pretty much from the in vitro fertilization, the reproductive technology currently used in humans, all the way to kind of mid-stage where placenta needs to be attached and supply the oxygen and nutrients. So that is all the kind of growing progression. And of course, the key here is understanding how placenta functions. So not only it will help to develop this technology to better optimize the oxygenators and devices, but also, while we still have natural reproduction, to address pathologies and to understand why some of the placentas fail. So the placenta, again, very briefly, is a critical life support system. It's the interface between the mother and her baby, and it's an immensely complex system with a massive number of scales and physical processes involved. And in the first part, I was talking about the maternal side of the placenta, the porous medium which it forms. And today, I will talk about the fetal side, the vascular network which formed inside the placenta and extends all the way to connect to the fetus, to the umbilical cord. So these trees, they host that vessels of the fetus. So again, I wouldn't talk too much about this, but I remind you about the structure because it's relevant. So that is the whole placenta, entire placenta, cast with resin. You can see umbilical artery and vein here, and all the branches. And if you zoom in, you could see the tissue envelope containing this capillary or larger vessels, and even at the finer scale, that's 50 microns. It's about half of human hair width. You can see that intricate capillary network, this green, which you can see cross sections below, right? And it's all hierarchy of scales from tens of microns to tens of centimeters. So it's in its immense complexity. And the challenge is to really try to get upscaled models which take into account this intricate structure but can operate at much larger scales. So again, the function is of course related to the structure very much here, and this is the interface, the actual interface. So inside you can see for scale the blood cell, red blood cell of fetus, and outside is a maternal blood, but you don't see individual cells, but they're never mixing, they're separate. But the nutrients, oxygen, glucose, other gases and nutrients, and also immune materials can cross. So somewhat, there was a question Isaacovich asked at the beginning. It does have somewhat a lymphatic system function as well as many others, but I only talk about the simplest transport, which is the passive diffusive transport or facilitated transport, but still without any energy requirement, without need for the active energy-dependent transport. So I wouldn't talk about that in detail, but only remind you, or those who first time tell you briefly, that it's always helpful to think about analogies. An analogy here is the blood-brain barrier. It's much more studied than placenta. And often the question of interest is to understand the transport of drugs across the blood-brain barrier, for example, for tumor treatment. And often it's quantified as how easy it is to cross in terms of solubility in oil or water, and the rate of crossing the barrier, permeability. And typically it's a linear relationship. The more it's soluble in oil, the better it is crossing. But there are some exceptions. For example, something crosses much less than expected because it's pumped out of these barrier cells, and something crosses much quicker than expected, like glucose, because there are some facilitation. Otherwise, it's very hard for it to cross. So our purpose of this is to try to get a motivation to build a similar picture for placenta, to understand how its structure and chemistry, combined with physics, can predict its function. So that's just a hierarchy of kind of historic development of the models from the very much black box that things comes in on one end and comes out on the other end without any appreciation of inner structure or spatial organization. Then some idealized models, and more recently, a very detailed 3D image-based, directly informed by the variety of methods, X-ray micro-tomography, confocal microscopy, and many other techniques. And the purpose of this approach is to try to really use directly very rich but highly complex information to distill it to more averaged, more upscaled quantities, which we can then see at a larger scale of the whole organ. So with that, I really stop at this part and jump to the other side. I think it will be easier if I'll just do it this way. So now we are going to consider the flow in the fetoplacental network. Okay. I think it's this. I think I need to jump to the beginning. Let me just restart. Yeah. For the delay. Okay, so what we're interested in this part
It's okay, we can see the second part.
Okay, so it's still loading.
Mm.
I think you can only see this slide part 2, right?
Yes.
Well, while it's loading, I'll just tell you briefly what you hopefully will see. Okay, it's loaded. Great. That's a really complex network. And we can use a variety of approaches to extract key geometric features, such as the connectivity of this network, like the central line, and also to get the understanding of the interface, such as the surface. So what you can see here is just some trajectories of individual particles, if you wish, and they can, of course, diverge quite widely along this network. And we can think of some models to predict the transport in this network. For example, Stokes flow, driven by the pressure drop, and advection diffusion, which you can see here. But note that there is an important factor here called the advective facilitation. And the reason for it, the presence of red blood cells, which are a significant booster to the advective capacity, because the oxygen binds very strongly to red blood cell. Without this presence, there's practically no chance of delivering oxygen in any quantity.
Excuse me, Igor, can we ask questions?
Please.
So looking at this figure, what you have and these trajectories, it looks like there are some loops, that this is not just a directed network of blood vessels going one way, but kind of something complex and not necessarily one way, but both ways, and with these cycles and circles and everything. Is it correct?
That's correct. Yeah. So that's no. This just if we think of that as a one-dimensional graph embedded in three-dimensional space, then indeed it is not just a tree-like structure. It's a network with loops, but at any for given boundary conditions, there is a unidirectional flow at any point. But indeed, it has redundancies, so to say, and I will talk about this later. And it's indeed very an interesting feature that it is quite complex structure. And that's one of the reasons why we want to think of some simple approaches to try to describe it, because each image, say computationally, to predict flow and especially transport for a variety of transport regimes, it might take CFD like 14 hours per sample. And if we have 4 samples, it's already very long. And also processing the samples is extremely time consuming. So that's not really sustainable if we want to understand placental function, because every placenta has some variability, and there are, of course, inter-patient variability, inter-variability between the normal and abnormal placentas of various degrees. So, of course, having some metric, some guidance in what we want to look into this system is effectively the primary aim. But for now, just what I want you to remember is that there is that factor B, which is effectively the chemistry of how much the solute in question, say oxygen, binds to hemoglobin inside red blood cell. And for some solutes like oxygen, it's of order 100. For carbon monoxide, which is a poison, why people die of suffocation, it's 10 to the power of 4, so it's huge. And for some gases like nitrogen, it's 1, so there's no specific boost from being able to connect to hemoglobin. And the other side of the story.
Excuse me.
Yeah?
Could you please explain? I don't understand about facilitation because, well, the velocity of red blood cells is the same as the plasma velocity, or nearly the same. Well, I know that average velocity of RBC may be high due to the concentration near the axis of the vessel, but well, the ratio of the average velocity of RBC and blood is maybe 2, but not 10 and 4. So I don't understand what is the physical meaning of advective facilitation.
Yeah, so thanks for asking this. I know I should have clarified. Indeed, so it's not to do with the velocity of the flow. Flow is, well, we assume that it's Newtonian here, of course, which is simplification, but we can we also revise that later. But this is coming from effectively a mixture theory. So there is a concentration bound to the hemoglobin sitting inside red blood cells, and there is a plasma dissolved concentration. And what this C really represents is the concentration of plasma, and there will be an equilibrium. And what this factor is coming from is, if we want to combine concentration, then what will it involve? It will involve the dissociation law between the bound and unbound form.
Yeah.
And if we effectively replace that C with the freely dissolved and bound form, then it will come as C plus C times the amount of binding, the kind of the dissociation law. And if you put it through the gradient, it will result in the derivative of the dissociation curve. Yeah, sorry, I probably know a bit hand-wavy. I know I could refer you to the paper which explains it in much more detail. But the idea that it really all down to the fact that there is the equilibrium between the bound and unbound form, and this B is literally approximating the gradient of this dissociation slope, how quickly you reach saturation of this, what's the maximum capacity each red blood cell can carry. Literally.
So we can think about two kinds of say oxygen, bound and unbound, and here we can forget about unbound oxygen because it is low concentration. Yeah. And
yeah, so
think about the oxygen carried by erythrocytes. But what is dP here? Is it a diffusion coefficient, or it is the effective Sher-induced diffusion coefficient?
dP here is a diffusion in plasma. So because red blood cells are so large, they really don't, their diffusion is negligible. So the only diffusion we can care about is diffusion of oxygen in plasma. So that
I see.
But in a sense, they are right. It's a good question to do it carefully, but that's been done several times, and it's literally a linearization of a nonlinear dissociation curve. So B is a slope here, which is 1. Then you have no red blood cells or nothing binds to them, and then it can become very large. If effectively everything is bound to red blood cell, then it becomes huge.
Okay. Yeah. Thank you.
But thank you for asking. I think it's good to clarify that. So the second component of that model is the interface, and here we have, and if I would be in person, I'll probably bring that 3D print of the network and show it to you. But you can see it on the image. So that is effectively a shell, right? Or envelope, the blue one, and the red one is a network, the capillary network, and in between is the tissue. Just connective tissue or some cells. And of course, any oxygen or other solute has to cross that barrier. And what is helpful to think about is if we, for example, ignore the metabolism, any reaction in the tissue, which actually come back, hopefully if time allows, then it's just diffusion in the tissue and nothing else. So we only prescribe a concentration at the interface and what we deliver, for example, from the inlet to the network. And then we want to know, how much crosses over that interface. So we solve the diffusion equation subject to that fixed Dirichlet boundary condition and for very, very complex geometry. So challenge here, of course, not in physics, but in geometry. And what it turns out that everything can be characterized by just one length scale. So if we think of the flux across the entire interface, then it can be just characterized as the diffusion. This time it's in tissue, dt. The difference across the interface, which is we assume constant. So it's a difference between the inlet concentration, for example, 0 if there's no oxygen from fetus, and maternal one, so maternal minus fetus, times a particular length scale, which is effectively an integral quantity of the surface area and average distance and so on. So if we'll think of lots of physiologists know what's a fixed law, which you have if you have a plane interface, then all you care about is the ratio of the area to the thickness. But for a more complex interface like that, it will be just a quantity which can be extracted from a computation. So if we just solve that problem on that interface, we can identify what this length scale is, and it will be unique, of course, for each sample. Okay, with that knowledge.
May I ask a question?
Yeah.
For the previous, yes. We have. What are the boundary conditions? We have the concentration on the inlet, yes, and concentration on the maternal interface. And we want to know the concentration on the outlet. There are...
That's ultimate question, but this particular problem, we want to know ultimate maximum flux we can deliver. So in this case, what we can say that we prescribe the 0 concentration everywhere on the fetal side of the interface and a given concentration at the maternal side of the interface, and see how much we can carry if the flow will be extremely fast in the network.
So we have concentrations.
The concentration in the maternal part of the interface is constant, yeah?
Yes, that's right. And likewise in the fetal side.
So we want to know how much of this concentration the fetal interface can grab from the maternal interface.
That's right, yeah.
For the inland blood flow. Fixed.
Yeah, but in this case, we want to have a limit. So what's if we produce, if the flow is as fast as possible, so if it flushes everything immediately, what's the maximum flux we can deliver? So it's a flux which we wanted to know, not concentration. So the flux is literally the integrated diffusive flux across the interface. So, and that will be limited by just effectively the average thickness and shape of your interface. Literally, it's all that influences that flux. So for
no question. The flux it is blood flux or flux?
Oxygen flux or any other solute.
Concentration of oxygen.
Yeah. So it's diffusion of solute across. So there's no blood in the tissue. So and for this purpose, we don't care about what's inside the capillaries. We just want to say that it grabs anything it comes to it, and we give maximum supply to the maternal side, or a fixed supply. So what's the maximum? How quickly we can get stuff across? Exactly, that is the question, and that is characterized by this flux. So it's total flux. It's measured in amount of solute per unit time, literally.
Thank you.
And the T here is the diffusion coefficient.
Yes, in tissue, inside this, in between blue and red region. So
which is known, yeah. So
it's a good question, which we can guess to the best of our ability. Yes. But generally, we would expect.
So from here we can guess about L, the distance, the characteristic distance L. Yes.
Yes, L is our target here. Yes. But dt, well, if it's a nice simple solute like oxygen, we can just say that diffusion in plasma is probably the same as diffusion in tissue. But for some complex solids like glucose, it's certainly less trivial.
Okay, yes, thank you.
Okay, so the good news are that it turns out that only 3 parameters, 3 geometric quantities are needed for quite comprehensive characterization of the transport in the exchange across that unit. So one I just described is the this kind of maximum exchange capacity or diffusive exchange capacity, and that's literally just the length scale, which again, for the simplest, if you think of the simplest possible interface, just the 2 parallel planes and one maternal, one fetal, right? Then it's all will be just the area over the distance. That's what this length scale is, right? So the larger the area, the thinner the interface, the more you can the more you can deliver. For for complex interface of that, of course, it's less trivial. Then there is more resistance metric. So how easy it is to actually push flow through the capillary network. Again, it's a function of a network. And then there is some just reference length scale, say total length of all the pathways through the network, or the longest pathway, for example, or the typical pathway. But anyway, it's just a reference. And all these 3 parameters, it turns out sufficient for quite accurate predictions. But before we go into it, I will say how to do that. So, and then we need to identify some non-dimensional groups, some parameters, and there are 2 of them here. One is the barrier diffusive capacity, so how easy it is to cross the barrier effectively, and that's literally characterized by the ratios of the diffusion in tissue versus diffusion in plasma, and also how large is this diffusive capacity, right? So how large surface area relative to typical thickness, and so on. And the second one, even more important, is the so-called Damköhler number, which compares the flow in a network to the transfer across the barrier, right? And it has, so note that there are 2 colors here. Red corresponds to geometry, and blue corresponds to physics or chemistry. So for that comparison of network advection rate relative to the transfer across the interface, the barrier, then it's characterized by the how fast you pump blood through the capillary network, so it's relative pressure. Also, how much it's facilitated by the presence of red blood cells, which can bind solute, relative to the diffusion in tissue. But there are 2 length scales, parameters, which both relates to length scales, one of the interface and one of the network, but they're purely geometric. And all this gives us a number. Okay, so that again, just keep this in mind for a moment. So what we can do, we can run CFD, which I said very expensive, because it's a full 3D, very highly complex geometries, complex meshes. It takes 14 plus 12, 20 hours to run the parameter sweep of that kind in each of the samples. And you can see they all look different, right? So it's taken actually from the same placenta at slightly different conditions of fixation, but they all look different. And what we would expect, that as the flow through the network is very fast, as I said, is we push it really fast, then it becomes limited by diffusion in a barrier, and we call it diffusion limited, and it reaches some plateau. So we reach this maximum uptake, which I showed previously, which is proportional to that that length scale L, curly L. Okay, so that will be some constant, but it will be different for different samples because it depends on the size, surface area, and so on. Now, if there is very slow flow, then the limit actually in the flow, so it diffuses relatively quickly, but then it doesn't really removed, so it's saturated, right? And in this case, it's Poisson-like resistance which limits the uptake. Okay, so if you have no flow, there will be no flux coming from outlet, right? And of course, somewhere in between the transition. But you see, it's log-log scale. It's an order of magnitude difference, both in the lower flow and high flow regimes. So the question, can we actually get a universal description? And the reason I'm presenting it to you today, the answer is yes. And if we use the parameters and the right parameter groups, then it all collapses. And all this complexity disappears. So you can characterize very different geometrically samples in a very concise way. And I'll explain you what these larger symbols mean. But you can see that we converge these limits. And even more helpfully, not we just can collapse it in terms of presentation form, we can actually find some asymptotic approximation, because certainly for Poisson flow, it will be just proportional to the flow rate, the flux in a capillary network. And in that regime, it will be proportional to the, just a constant, it is a constant. And also there will be some interesting features which I'll mention later about boundary layers. But the point is that we can get a composite asymptotics, which effectively link, interpolate between these two extremes, where everything driven by the flow and everything driven by diffusion, which depend just on two of these parameters, the Damköhler number and that capacity, mu. And in some sense, this one is not a significant effect. So even one parameter will capture majority of the effects. So for very large inverse Damköhler numbers, you just reaches the maximum, just constant. For very small, it's approximately linearly scaling, inversely proportional to Damköhler number. Okay, and now we can put this together. Right? It's not, of course, everything, but certainly it's quite comprehensive for microvascular exchange. And because we included chemistry explicitly, we can look at any solute which obeys kind of energy independent transfer. And we can say, okay, so we have different regimes depending on these parameters. So we can have diffusion limited for very strong flows. We can have flow limited for rather weak flows. So flow is a limiting factor. And also there are some more exotic regimes where you have very, very, very thin barrier. Then you can develop boundary layers. And then transition can be through the additional regime. But I wouldn't talk about this much, but please ask me if you want to know more. And that for symbols are our four samples. And they sit quite nicely in terms of at least the diffusive capacity. And also, which was surprising, they also separate in terms of advective capacity. And that wasn't obvious from images, but they indeed were fixed at different pressures internally in the capillary network, which means that they effectively have different integral resistance and possibly a little bit different surface area. So we couldn't see it from individual metrics, like when we look at the radii of these segments in the network and so on. But once we put it together as a Damköhler number, we can see the separation. And as I said, we can start looking at all kinds of solutes. For example, for oxygen is a reference. We have kind of that regime where it's just transition into the diffusion limited. For glucose, it is quite diffusion limited. And for some gases, like, for example, which used for anesthesia, it's quite flow limited. And you can see a huge range of scales. So it's order of magnitudes. Again, it's log-log plot. And likewise, you can see the separation in the diffusive capacity, how easy it is to cross. So for glucose, it's relatively hard to cross. And this is mannitol, which is a small drug, or fructose, which is similar, and it's very hard to cross. But what is interesting that if you even focus on the gases, for example, oxygen, CO2, and the carbon monoxide, this Poisson I mentioned, then they all have the same diffusive capacity, right? They're all sitting here, but they separate entirely according to the advective capacity. And indeed, if you look at the CO, it's literally, you can almost use it as a measure of the diffusive capacity, because it binds so strongly to the red blood cells that it's flushed immediately out of the network, and it takes as much as it can, right? So which is kind of why it's so dangerous. It competes with oxygen. Okay, so and that really helps us if we want to compare the performance. For example, we can say, well, if we look at the for example, ethanol, which it crosses the placenta, but it crosses at relatively flow limited fashion. So that transfer, if we follow that isoline, is comparable to the transfer of fructose, which is also crosses, but quite slowly. But they cross slowly for different reasons. Fructose crosses slowly because it's large and it has to cross the barrier, right? That's why it's end up here. While ethanol, it's flow limited because it doesn't bind to red blood cell. It doesn't have advective boost. So their transfer rate is somewhat comparable, but for very different reasons. So that really helps to appreciate that, to understand the exchange. It's not enough to know structure. It's not enough to know flow, but really it's the integration which allows that appreciation. So if you want to optimize some performance, then you really want to move in the gradient of that kind of exchange flux, uptake flux, which is somewhat diagonal to just the barrier properties or the flow properties. So that's one point. And another point is, indeed, we wanted to know how much it matters the topology of the network, or does it matter if we neglected the non-Newtonian nature of the blood? And it turns out the network is quite robust. For example, we can knock out a few of the branches and it doesn't change the net uptake. So it's certainly quite a bit of redundancy in the network. And also if we use, because it's a 1D network, so that was another simulation we used, just in a 1D graph where you can run the simulation. It compares quite well with the 3D within reason, and it doesn't matter if we use a Newtonian or non-Newtonian for the purpose of predicting the uptake. So at least at that level of kind of description, it doesn't seem to be important, but it remains to be confirmed, and that's actually what we want to know next. Is does the rheology matter in some situations and not in others? And there's other thing which is coming to the very first question: why is it loopy and does it matter? There are very interesting things if one goes sub-network level. So let's take just one sample, and we can color some interesting segments. You can see that there can be some kind of counter flow arrangements. So there's a inflow and outflow, and they can be very close. Right? So you can flow from one place and flow into another place where you kind of expect high concentration get nearby very low concentration. And if you plot individually kind of per segment the uptake as a function of flow rate, global flow rate, then it turns out that not all of them are monotone. And the reason for that is because one of these kind of donor-recipient mechanism could kick off. So where you have two countercurrent segments, then the segment which gets in oxygen at the end can start getting it back to the inlet, which is very poor in oxygen, before it leaves the network to larger kind of arteries and veins. Sorry, to venous side. So if I would say we look at the blue and the green, so that you can see that the blue is the inlet part and the green is the outlet part, and the blue here effectively acts as the receptor, and the blue and the green, as a donor, because it already gets all the oxygen and getting out of the system. So that's why I get a transient rise. But because of the network size effect, these will integrate out. So even though we can have these, curious effects and a kind of counter current loops, if you look at the whole loop, it will still be monotonic for sufficiently large network. So that I think is an interesting point. So it depends on the question you ask. If you do surgery or something at the level of one capillary network, then it's important. If you want to predict function of the organ, then probably it's less significant.
Now, Igor, but still I don't understand why this complex geometry. Is it just because these blood vessels are not developed well enough, like it happens, for example, in tumor growth? We know that there are also such underdeveloped blood structures, or there is some purpose, if you can say so, hidden behind, and it really helps something.
It's a very good question, but I don't think I have a good answer. Well, there are multiple kind of competing suggestions, but all of them are hand-wavy and physiologically kind of arguments rather than mathematical or physical arguments. One, the placenta is rapidly growing organ. And in some sense, what it does first, it just massively develops. And somewhat many people do compare placentas to tumors, but in a kind of control, which are developing in somewhat a controlled way. But they develop very quickly and very aggressively, and they often overdevelop vasculature, which then is pruned. And then it's what you're left with. Other question is, I think what's important for placenta is to have very low resistance, and of course having multiple parallel paths is very helpful. And also the structure is fundamentally three-dimensional. So you have to have this actually bulb surrounded by the maternal blood. So it needs to be well perfused and close to the periphery. So there is also a question of having the segments close to the surface versus segments inside, kind of far from the surface. So there can be effects of diffusional screening, which I think somebody mentioned last time. So but I don't think any of them are leading hypotheses. So and also it's dangerous sometimes to ask question about what is effective or efficient, because the placenta is so divergent that it somewhat works for its own species, but it looks different in different species, and it doesn't mean that one placenta is worse than the other. It just does its particular job for a given purpose for a given time, and it's very transient organ. It's growing constantly. It's never static. Does it answer the question?
Yes. Okay. Thanks.
Right, but I'm conscious of time, so I'll try to press on. There's not much left, but I'll. So I mentioned that I, to start with, ignored the metabolism. So we just have a passive transfer of solute through this barrier for the tissue. Now, well, it's not the case for all solutes, and actually for many solutes. And for example, briefly, I'll mention that placenta is unique in a way that you cannot easily get a human lung and experiment with it. You can with human placenta easily because it's a human waste. And because I'm based in Manchester and we have a center which has 9,000 or 10,000 specialized deliverers per year and a dedicated research center, then we have ready access to placentas, and we can put it in quite a complicated machine and perfuse it, kind of keep it in a closed physiological condition. So plug it in from the kind of fetal side and put some supply to the maternal side, like you see here. So there are kind of semi-closed circuit from the maternal side and a closed circuit from the fetal side. And we can put oxygen if we want to, and so on. And we can measure oxygen. That's most useful, and that was most recently done. So we can directly sample oxygen in a placenta. Sorry for somewhat clinical images, but you can see the kind of needle, the optode, which can sample placenta in different locations, and that is the canal which delivers oxygen. And we can build, for example, 3D maps of oxygen, like people do in soil for oil purposes, and it's porous medium of similar kind. And we can also quantify the uptake. So what we done here, we just delivered some amount of oxygen, reached steady state, and switched off the flow. So we expect oxygen to drop, of course, because it's been consumed by the tissue. And the rate of this drop will tell us something about the metabolism of the tissue. So we can fix it, fit it to some simple lows like sigmoidal-like saturation low, and get the maximum metabolic rate and also the kind of half saturation constant. So again, it's not perfect and it's somewhat a pilot experiment, but it shows already quite some variability between different. So this shows different placentas, by the way, so different colors, and they have already, you can appreciate different time scales, even though of some in presence of some noise, of course. And we can extract these parameters. Now, if we compare the placenta to other tissues, for example, to the brain or liver or heart, then we can see that the rate of metabolism is comparable, although might be a little bit lower than, say, in brain, of course, which is understandable. Brain is massively oxygen dependent. So placenta is not as oxygen hungry, but it still needs oxygen. But its half saturation seems to be quite different, so that's interesting. But it's not confirmed because it's somewhat pilot. So that might mean that there will be different type of kinetics and kind of slower to reach, slower to reach the saturation than other tissues. Okay, but what I want to tell you about mathematical approach to that, and all which we change is we just change the model inside of the tissue between the outer shell and the network, right, in the barrier inside. And we have this metabolic rate parameter, which I just kind of called actually Q previously, but it's related. And again, in the interest of time, but I'm happy to talk about this later, or give you a paper if you need. There's another parameter which needed, which compares the metabolism to diffusion and to the surface area, and again this diffusion length scale. And that parameter is sufficient to describe the impact of metabolism. So you can see that if it's strong, we expect boundary layer developing now in tissue. While if it's small, then we can really ignore it and it'll be just diffusion. And again, we can look for different solutes, and it turns out that for majority of them, actually, it's not huge. It's more or less negligible for oxygen and somewhat for glucose. But for fructose, it is more significant, especially because it's somewhat dependent also on the baseline concentration. So what's the concentration in outside the barrier, the plasma? So again, what I typically tell to clinicians, if mother drinks fizzy drinks, then it means that the uptake of fructose by placenta becomes significant. So, and that cannot be ignored. And if we think about our parameter space of kind of fate of the solute, then we now have 3 dimensions: the advective capacity, diffusive capacity, and metabolic kind of parameter, or also capacity. And if we now focus only on the advection and metabolism, all right, and now then quite interesting thing happens. So, well, that's kind of how it looks like now. That's numerical curve. If you focus just on that line, so again, it's flow limited, diffusion limited, but that boundary start to bend for increasing metabolic parameter. So if I am a diffusion limited solute, say fructose, I'm sitting here, and as I increase metabolism, I suddenly find out myself in a flow limited regime, which is not totally expected. So you would expect that if it was limited by diffusion, why flow started to matter. So that wasn't intuitive. But some way to develop an idea of why that happens is because of these 2 regimes, very conceptually can be described. Again, thinking of the simple barrier, you have maternal concentration on the left, fetal on the right. If it's diffusion limited, then they're just fixed concentration, right? So 0, you can't get lower than that. And it's all driven by the gradient across the barrier. So you just read this gradient, and that's how you find out what flux you deliver. And as you increase the uptake, metabolism in tissue inside the barrier, then this gradient, of course, will diminish, but not as quickly as in the case of flow limited, in which case we literally, because it's limited by flow, then we effectively always trace the concentration at the fetal interface. Because the flow is very slow, it has time to diffuse through for any kind of choice of uptake or flow. So for slow flow, it always will be constant inside the fetal network. And then all you care about is how fast you can get this concentration away. So concentration times the flow rate, that will be the flux. So in this case, what matters is the concentration you detect at the fetal interface, and that will depend much more strongly on the uptake, because the concentration will drop much quicker than the gradient, as you can see from these cartoons. So that really kind of qualitative hand-wavy explanation, but we have more quantitative way to do it using the WKB asymptotics, because they're both of them exponentially small as you increase metabolism. But it turns out that indeed the flow limited becomes much smaller than diffusion limited for strong metabolism. So it does become a limiting factor for strong tissue metabolism, which is totally unexpected, but that's what it is. And just to summarize, so fortunately I'm on time, what I wanted to show you is a way to really manage the complexity of a system which is just a part of a much larger organ. And it turns out that we can have quite a robust system of parameters which comprehensively characterize the uptake. And actually, even the asymptotical curve I showed you is within 30% of kind of inaccuracy at the at the most difficult regime, far from strong flows or weak flows. And it should be applicable not just to placenta, but to many other systems which have the vascular side and tissue side, which need to transfer and exchange things across. So I think lymphatics actually one of the candidates. So we might be able to talk to Jimmy Moore at some point. And once we know what determinants to look at, so we know what really matters, then the next step, of course, is to try to look at these metrics and different pathologies and try to understand, what fails there and how we can fix it. So with that, I stop, and these are the references. So the key one are these top two, the interface focus and science advances. And I'm very happy to answer any questions. So thank you for your attention.
Thank you very much for this very interesting and stimulating presentation. Please, if there are more questions from our audience, then we can discuss them. So while our participants are thinking about their questions, so when you show this, well, it was somewhere in the middle, this parameter L, which characterizes this network. So it's not just the length, not just the surface area, it's something more complex, right? It's certainly proportional to the surface area, but not exactly. It's right. It's a probably kind of empirical parameter, which is not maybe straightforward to determine directly from the geometry of this network, right?
Yeah, I absolutely agree. But the powerful thing about that, this parameter can be totally determined by geometry. So once we know the sample, we don't necessarily, because the often hard part of all modeling of that kind is to run a CFD in the network, because that is always a limiting part in terms of numerical cost. While computing the diffusion, even in a complex interface, is much cheaper. So, for example, and even if image imperfect in many sense, so that, it's hard to quantify it. It's much easier to extract these quantities than extract the kind of the full quantities. So that is one strong point. So even though it's hard to predict it from just looking at the images, it's certainly much easier to predict that numerically. And also what we can ask is, in some sense, we can ask how much this varies. So we can try to correlate this, because we can measure surface area, we can measure average thickness, and we can start building correlations between the different geometrical features.
Yes, and also, well, when you say that it's okay, well, complex and long 3D CFD simulations, yeah, I totally agree if you consider such a complex geometry. But is it really necessary to consider the 3D simulations? Cannot it be replaced by 1D flow?
Well, we tried that, and it turns out that resistance is very important.
But you can introduce it. Yeah, sure. Sorry, please.
Yes, but you're right. And what we showed, I think, if I go briefly back to that picture, that literally looked at the 1D model, and we showed that it has a good agreement, but there are regimes where there will be deviations.
Now, so this is, yeah, sorry. I don't want to interrupt you. Please continue.
Yeah, so what I wanted to say that ultimately the power of the approach is that we don't need, so in some sense, we don't no longer need 3D simulations once we know what to look for.
No, I understand that. You justify, well, like you validate your approach with this complete modeling, and you obtain kind of simple answers. Now, this is very nice. But my question is about an intermediate scale between these complex 3D simulations and just a simple answer in terms of these dimensionless parameters, because when you show this 1D network, it really makes me think about what is called, and Anastasia wants to ask a question, I don't know about this, about different, but this is what she does about this 1D hemodynamics, or the same or similar models are used also for lymph flow. And in fact, they work very well, in fact, for this complex networks of blood vessels. They give a quite reliable method to study blood flows. So I think it's really an interesting question. So sorry.
Just one small comment I think here is that the main challenge here, indeed, in terms of just the flow network, we can get some estimation. We'll get resistance wrong, definitely, because it's highly complex, irregular, but we can get some flow properties. The challenge is in the transport, because of what is important, that they are coupled through the tissue diffusion. So we need to actually embed this network in the...
Right, yes. I think it's very interesting, and I think it might be also interesting to indeed to complete these 1D hemodynamic models with this diffusion through the bone. Well, to couple them with diffusion, I don't know whether such models exist or not. I'm not sure. I'm not very much familiar. But if not, I think it would be really a good extension of such models. Anastasia, please, you have a question.
Thank you. I would like to ask the question. You said that placenta is a fast-growing organ, and as I understand, the calculations are done with constant geometry. What changes in results may be, and can we consider the changes in geometry to model growing placenta in the real world?
Thank you. It's a very good question. I think the brief answer is who knows. But the extended answer is that all I showed, and which I should have been really clear, is for fully developed placentas and normal placentas. So once you go into realm of placenta, stage, which is so it's placenta which is delivered and which is normal placenta. So it's fully developed, fully formed. So it's changing over time. But the problem is that I said that it's easy to get placenta because it's literally clinical waste. So people are throwing it away. But hospitals and universities has agreement, and we have access to it. But it's easy to get placenta which is fully developed, or sometimes pathological placentas which are small and pathological. But getting somewhere in between is very tricky, because of course you wouldn't get placenta in a normal pregnancy before it's delivered. Yes. So that is a real challenge. So knowing actually what's going on, it's extremely indirect. And the main problem here, and why actually modeling is so important in this field, that no other placenta is really relevant apart from human. So so upside of human placenta, you can use real placenta at term and at delivery, but downside, you cannot use it at any other point. While at least you can use animals for lung development, you cannot use it really for placenta properly, because the lots of features are very different.
Thank you.
But also to complete what Anastasia asked, I think that even if not but what you say, that placenta is growing and changing, but I think that these are different time scales, and kind of a quasi-stationary approximation should be applicable here. So it changes, but these changes and growth of placenta probably are much slower than these exchange processes and flow and concentration distribution, all the things. So I think that quasi-stationary approximation should be applicable here.
Yeah, you're absolutely right. Yeah, so we're talking about probably 10 seconds or minutes for exchange, and weeks for appreciable change in the structure.
Thank you.
Are there other questions from the audience? if not, maybe I can ask another question. So, Igor, you mentioned several times that placenta are very different in different species, in particular in humans and other animals. So, but what is the most essential difference? Is it geometry of this vessels, or what? How they can be? Maybe the same parameters can characterize, in fact, placenta in different species. The values of parameters can be different, but maybe the characterization is similar.
Yes, I think the answer is that it depends on the animal. So some animals are slightly more similar than others, but I think the even arrangement of that the fetal capillaries are almost in direct proximity to the maternal blood, which is freely circulating. So there's no direct capillary network on the mother side of the placenta, or on the maternal side of the placenta. That is not the case for many animals. So many animals has placenta, which is literally more traditional vascular systems, where you have capillary side on the fetal side, capillary side on the maternal side, and then there's kind of porous medium on one side and a network embedded in this porous medium.
Oh, yes, this is very interesting. Yes,
that's a change, and also the speed of gestation is so different. So in mice, it's a couple of weeks, literally, and also the so I would say different animals have different features which are similar or less similar to, depending on what you look again. So I think the answer to that would depend on the question. So if we are looking for exchange of at the interface, it's one question, but there are, for example, models for pathologies, for example, building high blood pressure, which often happens in pregnancy. That's another pathology, and that's again quite unique to humans, which doesn't happen in animals really, or at least not only might be in some very higher monkeys, which hard to experiment with. So it's mostly geometrical difference, just to summarize, but also there are functional differences even down to the cell biology.
Yeah, it's so complex and well organized, well functioning system. Is it used for biological engineering or not yet?
Well, I mentioned
like kind of filters, or I don't know what.
Yes, well, in some sense, placenta is a biologically built, wonderful incubator, which effectively does the job of supplying solutes, clearing the waste, filtering out the waste, providing water supply, energy supply in terms of heating the blood if necessary, and many other things. One of the possible interesting questions is, if we understand well how placenta works in the human, because what's important to understand also regimes and what kind, because since you've seen many solutes, it's impossible to have all of them optimal. In placenta, I think often what we try to think about, we're not trying to think about optimality, we think about robustness. So rather how much you can push it before it fails, because each solute will have its own optimal regime, and it's impossible to jiggle all these many solutes. But for example, we work with people who develop artificial oxygenators. So it's not in the kind of what I showed in this kind of lamp in a bio bag, but who want to develop oxygenator which pushes blood by own heart of the fetus, of the prematurely delivered fetus, to help them breathe effectively. And understanding properties of placenta will help us design better artificial placentas. So that's another kind of line of work. So we need to what we're doing, we're reverse engineering it.
Okay, guys, thank you very much again for this very interesting presentation. If it is possible, I would like to ask you to send us your slides, and I mean, if it is not confidential, then we will put it in our seminar website, as we usually do, and then people eventually can consult them, if you don't mind, of course. Okay, thank you very much.
Thank you
very much again. It was a pleasure to meet you here, and I hope you will come from time to our seminars, like as we did for the Moore seminar, for example. So I suppose that you receive our announcements now. Yes. So thanks again, and thanks to all our listeners, and then we'll meet probably the next week. Анастасия, что у нас планируется дальше? Вы можете нам сказать?
На следующей неделе, это лучше у Кристины спросить по аспирантскому семинару, а следующий общий семинар у нас будет 3 июня, если я не ошибаюсь, выступает профессор Бертолио. Я пока не знаю, с какой темой. Да, но у него что-то с МРТ связано.
Бертоглио, по-моему, да?
Бертолио.
Да, я его немного знаю, по-моему, когда-то я, по-моему, слушал что-то, он занимался, по-моему, моделированием аневризмы, а сейчас не знаю.
Ну, у него там несколько проектов, так что...
Но в любом случае это будет интересно, так что, Игорь, мы вас также приглашаем послушать.
Спасибо. Да. Получится, не будет никаких других течений.
Ну что ж, хорошо, всего хорошего, всем спасибо, до свидания и до следующих встреч на нашем семинаре. До свидания.
До свидания. Спасибо большое. Спасибо, до свидания.
Спасибо, до свидания.
До свидания.